everyone. Today we'll discuss the part 57 of the important notes for biochemistry. SGOT or the serum glutamic oxaloacetic transaminase, also known as aspartate transaminase, this enzyme is present in the mitochondria, while the SGPT, that means serum glutamic pyruvic transaminase, also called as alanine transaminase, that is the ALT, is present in the cytoplasm. Regan is an isoenzyme of the placental alkaline phosphatase which is heat stable and expressed in some cancers. Hemoglobin has buffering action due to the presence of histidine. Histidine has maximum buffering capacity because the pKa value of the imidazole group of the histidine is very near to the physiological pH. Carboxyn is a fungicide which is inhibitor of the complex 2 of the electron transport chain. Cysteine proteases, also known as thiol proteases, are papain and the caspases. Carboxy proteases that hydrolyze the peptide bond at the carboxy terminal are the pepsine and the lysosomal catepsins. Metalloproteases are the carboxy peptidase A and the carboxy peptidase B. Metalloproteases are the exopeptidase, very important. Acute phase proteins are a class of proteins whose plasma concentrations increase as in the positive acute phase proteins, or it will decrease as in negative acute phase proteins in response to inflammation. This response is called as acute phase reaction, also called as acute phase response. This occurs in response to the inflammation or tissue injury. Now the negative acute phase proteins, as we discussed before, the production of these proteins is decreased. These are therefore referred to as negative acute phase proteins. Negative acute phase proteins are albumin, transferrin, transthyretin, transcortin and the retinol binding protein. Positive acute phase proteins. These acute phase proteins are the C-reactive protein, D-dimer protein, mannose binding protein, alpha-1 antitrypsin, alpha-1 anti-chymotrypsin, alpha-2 macroglobulin, fibrinogen, prothrombin, factor VIII, that is the anti-hemophilic factor, von Willebrand factor, plasminogen, complement factors, ferritin, serum amyloid P component, serum amyloid A, ceruloplasmin and the haptoglobin. Methods to detect the protein-protein interactions, they are, first of all, it is the biochemical methods. Over here, immunoprecipitation, affinity electrophoresis, strep protein interaction experiment or the SPINE, proximity ligation assay, multi-mass spectrometry and the biomolecular fluorescence complementation. These all are the biochemical methods and they are in vitro. The next method, it is the biophysical methods. Over here, interferometry, static and the dynamic light scattering, plasmon resonance, flow-induced dispersion analysis, fluorescence polarization, fluorescence correlation spectroscopy, fluorescence resonance energy transfer, fluorescence lifetime imaging microscopy, fluorescence polarization, microscale thermophoresis, rotating cell-based ligand binding assay and the single color reflectometry are the methods over here and they are also in vitro. Next is the computational method. Over here, homology based tools are used and the tools are based on the structural classification of the proteins and this is in silico. Genetic method, over here 2 hybrid screening method is used and it is in vivo. Now a buffer is the most effective when the pH of the buffer is equal to the pKa of the buffer. That means salt is equal to acid into full ionization. When the pH is equal to Pi, that is the isoelectric pH, then a buffer has minimum buffering capacity. Effective pH range of a buffer, that is the range where there is maximum buffering capacity of the buffer, is equal to pKa plus or minus 1. Bicarbonate buffer is ideal because both the constituents of this buffer can be altered in the body. The bicarbonic acid, that is the H2CO3, is maintained by the respiratory system while the carbonic acid, that is the HCO3 minus, is maintained by the renal system. So its components can be increased or decreased by the body. Major extracellular buffer, it is the bicarbonate buffer, very important. Major intracellular as well as major urinary buffer, it is the phosphate buffer, again very important. So that's all for today. I hope you found this helpful. Thank you so much.